Hello everyone, welcome you all in this video lecture. So in today's video, we will discuss about the coding DNA sequences or exons. So let's start with the introductory part about the coding DNA. So in this video, we are going to discuss about the coding DNA sequences or exons in detail. So before moving to the topic, it's very important to know about some basic facts about the DNA and what are the different types of DNA sequences. So basically the DNA is named as deoxyribonucleic acid and it is the type of a nucleic acid. We know that nucleic acids are the important classes of macromolecules which are found in all the cells, in prokaryotic cells as well as in eukaryotic cells. The nucleic acids are first discovered by Friedrich Misser and it is of two types, the DNA and RNA. And the DNA acts as hereditary material in most of the organisms except some viruses and some prokaryotes, and where the RNA act as a hereditary material. Now the DNA occur in the nucleus of the cell. We know that cells are of two types. The first one is prokaryotic cell and the another one is eukaryotic cell. So in the case of prokaryotes, where there is not any well-defined nucleus, the DNA found in the cytoplasm of the cell. But in the eukaryotic cells, there is a well-defined nucleus which is covered by nuclear membrane, and the DNA found as a chromosomes in the nucleus of the cell. Chromosomes have DNA with histones and non-histone proteins. And DNA helps in all the cellular activities like replication, transcription and many other cellular activities. So basically the DNA have two types of sequences which are coding DNA sequences, also called as exons, and the another one is non-coding DNA sequences which are known introns. So in this video lecture, we are going to discuss about the coding DNA sequences, their structure and their functions in all cellular organisms. So let's understand and describe about the coding DNA sequences in detail. So as we have discussed that these sequences are also called as exons. And exons are the coding region of the gene. We know that genes are also found in DNA and DNA have different genes. So the genes contain the information which is required to encode the protein, because we know that genes are the hereditary unit in all the organisms. So what happens in eukaryotes? The genes are made up of coding exons with some interspersed non-coding introns. So the exons are those DNA sequences that have the ability to be translated into amino acids and protein chains. In prokaryotic organisms, there are not any introns, but in the case of eukaryotic genes, they have some introns interspersed in between exons. And introns are the non-coding sequences which are found in genes. And these non-coding sequences have not any ability to be translated and to synthesize proteins. So they are removed from the mRNA script by the help of the process known as RNA splicing. So here in this diagrammatic presentation, we can see the location of exons or coding sequences of DNA. So here this is the DNA stretch having intragenic spaces between the core elements, and they are the genes in between these intragenic spaces. So when we see a structure of gene in detail, so this is the gene having different introns and exons in its structure. So there are the blue colored regions which are indicated as introns or non-coding DNA sequences. So these introns have not ability to synthesize proteins, and in between these introns there are orange colored regions which are exons or coding regions of the DNA. So these exons are responsible for the translation and protein synthesis, and before the translation by the process of RNA splicing, these introns are get removed by this mRNA script. Now let's understand about the discovery of exons. So we know that the term exon was used to the coding sequences of DNA. And this term was first used and first coined by an American biochemist named as Walter Gilbert in 1978. So Gilbert termed the intragenic regions between exons as introns, and the regions which have the ability to be translated and which can be expressed as exons. Now let's understand the structure of coding DNA or the structure of exons. So the coding DNA molecules are made up of stretches of DNA. They are basically made up of nitrogenous bases or nucleotide sequences, and they have the ability to be translated into amino acids and proteins, because we know that DNA can function only with the help of protein synthesis, because proteins are responsible for different types of cellular activities, and they are also form enzymes because all the enzymes are proteinous in nature. So all these enzymes are responsible for the completion of different types of cellular reactions. So in DNA, they can be separated by introns and found as discontinuous genes. In eukaryotic organisms, they found as a discontinuous genes because there are introns present in between the exons. But in the case of prokaryotes, the introns are not present. So these introns have not any role in translation, and thus they are removed by the process of RNA splicing. And this process completes in the nucleus of the cell, and then the proteases completes in the cytoplasm of the cell. So here in this diagrammatic presentation, we can see the structure of gene and also the process of translation or protein synthesis by these genes. So in this structure, we can see a DNA stretch having different genes. So there are two genes in this DNA stretch, gene A and gene P. And in between these two genes, there are regulatory enzymes which are promoters. So in between these regulatory elements, they are called as core elements which have introns and exons. So here we can see in green color, they are the exons. And here in the red color, they are the exons or the coding DNA sequences. So with the help of transcription, when this DNA molecule is converted into mRNA, and after the mRNA script, here there is not a role of introns in the translation process. So this pre-mRNA script is spliced by the process of RNA splicing, and the introns are get removed from this script, and there are all the exons in the continuous manner. So these exons are further transported into the cytoplasm of the cell, and this mRNA script is further converted into amino acid script, and they are converted into proteins with the process of translation. So this is the process of translation or converting DNA molecules into the protein molecules with the help of translation. Now let's discuss about the functions of coding DNA and what are the functions of exons. So the main function of exon is to encode proteins, because these DNA sequences have the ability to synthesize proteins by the DNA molecules. And the protein domains can be encoded by a single or multiple exons, because there are several exons which can encode only a single protein like hemoglobin in eukaryotic organisms, and there are several exons which have the ability to encode for the multiple proteins. And sometimes the exon also allow for multiple proteins to be translated from the same gene. And this activity or this ability is called as alternate splicing of exons. So in this video, we have discussed about the structure and functions of coding DNA sequences.